Welcome to week 10 of Applied Immunology, the last week of lecture and discussion material for this course. We are going to cover two pretty different topics at the end of this course, starting with today's talk on mucosal immunology, then switching gears with lectures 10b and 10c, which will cover topics that are relevant to tumor immunology. Today's lecture 10a will introduce us to a set of specialized immune responses that are found specifically at some epithelial surfaces that are some of the first to interact with external pathogens, and these are called mucosal surfaces, or the mucosa. We will start off by defining mucosal surfaces, then discuss some of the interesting anatomical features, effector mechanisms, and regulatory mechanisms of mucosal immunity. We will then end today by talking about a concept called mucosal tolerance, which is essential for preventing improper immune activation targeted against commensal microbes or harmless food antigens that are found in the gut. Mucosal surfaces are defined as epithelial surfaces that secrete various forms of mucus, which are basically any epithelial barrier other than the skin. Mucus serves as an additional physical barrier by trapping non-motile microbes, which helps restrict their access to epithelial cells for infection. Mucus also contains molecules like antimicrobial peptides that can also help kill invading microbes. This figure summarizes the anatomical locations of mucosa in the human body. The reproductive organs and urinary system make up something called the urogenital tract, here on the left, shown in the green and yellow organs. The nose, trachea, and lungs make up the respiratory tract in blue, and the esophagus, stomach, and intestines make up the gastrointestinal tract in pink, which we also refer to as the GI tract or the gut. The mucosal immune system is made up of specialized lymphoid organs, cells, and molecules that are associated with each of these tissues. The epithelial surfaces that line each of these anatomical sites is often the first site where the body encounters infectious microbes, so we can view these as sort of a portal of entry for pathogens. The physiological functions of these epithelial sites, such as gas exchange in the lung or nutrient absorption in the gut, require for them to be quite thin and permeable, which obviously renders them physically vulnerable to infection. Therefore, the body needs to concentrate specialized types of immune responses at these sites in order to prevent infection and preserve tissue integrity. Today, we will particularly focus on mucosal immunity in the gastrointestinal tract, but please keep in mind that a lot of these features are shared at other mucosal sites as well. We've already learned about how adaptive immunity is primed at secondary lymphoid organs such as the spleen and lymph nodes. The adaptive responses of mucosal immune system possesses several unique features when compared to the spleen and lymph nodes. So let's go over a brief summary of these differences. We will discuss most of these in more detail throughout the rest of this talk. First, the mucosal immune system has a number of specialized anatomical features. These include the presence of lymphoid cells that are diffused throughout the mucosal epithelium, as well as the presence of organized lymphoid tissue structures that directly underlie the mucosal epithelium. There are also some specialized antigen uptake mechanisms that can be used to stimulate immune responses. Mucosal immunity also exhibits specialized effector functions that are only found at these barrier sites. These include the presence of effector T cells at epithelial surfaces that are poised to undergo activation even in the absence of infection. Mucosal surfaces are also the main place where we find IgA isotype antibodies. And if you remember back to our week 5 lectures on isotype functions, IgA is extremely important for defense against pathogens at these surfaces. Mucosal tissues are also unique in that they are colonized by commensal microorganisms, including bacteria, fungi, and viruses that we collectively refer to as the microbiota, or the microbiome. Because mucosal surfaces are populated with commensals, there is a unique requirement for regulating inflammatory responses. This is done through a variety of immunosuppressive mechanisms, including the presence of inhibitory types of antigen-presenting cells, as well as Tregs. This makes sure that inflammatory responses are not activated in response to the microbiota or to innocuous or harmless protein antigens, such as those that are found in food that travels through the GI tract. This is just a quick summary of some key metrics that we can use to characterize immune responses that are initiated at mucosal surfaces. We will learn more about each of these points throughout the remainder of this lecture. And again, in the following examples, we will focus on the gastrointestinal tract, but please keep in mind that similar or analogous features are found in other mucosal tissues as well. Let's start by looking at where lymphocytes are localized within the gut epithelium, shown here in pink, which is made up of epithelial cells that are also referred to as enterocytes. So far in this course, we've learned about how T cells are typically concentrated in lymph nodes, but that they can also exit and traffic to sites of inflammation following their activation by antigen-presenting cells in response to stimuli such as infection. Mucosal surfaces are an exception to this, since they are already seeded with lymphocytes. An important subset of these are called intraepithelial lymphocytes, or IELs, which are CD8-positive T cells that are distributed throughout the gut epithelium. The gut is also seeded with diffuse lymphocytes in the lamina propria, which is the name for the connective tissue that lies under the gut epithelium, and it's shown in this figure in light yellow. The lamina propria is populated with both CD4 and CD8 T cells, as well as plasma B cells, which produce IgA isotype antibodies. Importantly, there are also a lot of regulatory T cells, or Tregs, in the lamina propria, which are important for preserving this concept of mucosal tolerance. Note that although a lot of these cells have specialized functions that can act locally, these tissues still contain all of the same lymphatic vessels that we've already learned about, so cells and antigens can also drain to lymph nodes for traditional immune priming. In the case of the gut, the draining lymph nodes are called the mesenteric lymph nodes, and these are located in the connective tissue that holds the intestines together. Mucosal surfaces also have organized lymphoid tissue structures that lie directly underneath the epithelium, which we can kind of think of as new types of secondary lymphoid organs. In the small intestine, these include structures called Peyer's patches, which are structurally very similar to lymph nodes. However, Peyer's patches have higher proportions of B cells, which are interspersed with relatively smaller T cell zones compared to traditional lymph nodes. The gut also contains isolated lymphoid follicles, or ILFs, which are made up of mostly B cells. Together, the Peyer's patches, ILFs, and mesenteric lymph nodes are collectively referred to as gut-associated lymphoid tissues, or GALT. The GALT make up a massive physical portion of the immune system, as the intestinal mucosa alone contains more lymphocytes than are found in all other lymphoid organs combined. Because some components of the GALT are positioned within the intestinal wall, both IELs and the GALT are very important for rapid adaptive immune responses to signs of infection. Since these structures are predominantly made up of B cells, so things like Peyer's patches and ILFs, these structures are important for the activation and maturation of B cell responses. Meanwhile, the IELs that are found throughout the epithelium can mediate some important direct cytotoxic effector functions, which we will discuss next. Intraepithelial lymphocytes, or IELs, have some unique activation properties compared to naive CD8s that are primed in lymph nodes. So these IELs have an activated phenotype even in the absence of infection, so we can think of them as kind of being primed to respond to infection on short notice. There are two different subsets of IELs which differ with respect to how exactly they function. We can first look at type A IELs, which function very similarly to conventional T cells. These cells behave similarly to effector CTLs that we've already learned about, so they recognize antigen on MHC class I through their TCR. Upon activation by peptide MHC through their TCR, type A IELs then release granzymes and perforin to kill the infected target. They can also kill cells using fast ligand expression, shown here in the diagram, and this will induce extrinsic apoptosis, which we haven't talked about much in this class, but is just another mechanism that CTLs can use to kill infected targets. In contrast, type B IELs function similarly to NK cells, which we learned about in the beginning of this course. Type B IELs express NKG2D, and so therefore they can recognize the ligands MIC-A or MIC-B expressed on the surface of stressed or altered cells. Type B IELs can kill stressed epithelial cells using perforin and granzyme, but in contrast to type A IELs, this is done in a TCR-independent fashion. Again, this is instead mediated through the activation of NKG2D. Both categories of IELs therefore help kill epithelial cells that are likely infected by viruses, either through the presentation of cognate peptide on MHC class I, which is the case for activating type A IELs, or through the detection of stress-induced self to activate type B IELs. These types of immediate cytotoxic responses are important for things like viral infections that are initiated in the gut, because they help control the infection early on before it can disseminate to other sites throughout the body. Mucosal surfaces also have specialized mechanisms for sampling soluble antigens from the hollow space, or lumen, that faces the mucosal epithelium. Importantly, the way that antigen is transferred to APCs can change whether these cells undergo full activation to stimulate adaptive immune responses, or if the APCs undergo tolerogenic activation to promote mucosal tolerance. We will discuss how these things work a little bit later on at the end of this lecture. Let's begin by first going over the different ways that antigen can be sampled from the intestinal lumen. First, there's this process called transcytosis, where antigens are endocytosed or phagocytosed from the luminal side of the mucosal epithelium, and then sort of carried through the cell and then kind of spat out on the lamina propria side. This side is also called the basolateral surface, since it faces the basement membrane of the epithelium. Transcytosis is summarized in the left and middle panels here. On the left, specialized cells called microfold cells, or M cells, transcytose antigens to the lamina propria. In the middle panel, enterocytes will transport antigen-antibody complexes from the intestinal lumen to the lamina propria, and they do this using something called a neonatal Fc receptor, which is also abbreviated as FcRn. On the right panel, we have a third example of antigen transport, where enterocytes infected with intracellular pathogens will die by apoptosis. In this case, dendritic cells in the lamina propria can phagocytose intracellular antigens for processing and cross-presentation on MHC class I. Importantly, this specific form of antigen acquisition by dendritic cells is associated with an active intracellular infection, rather than just generalized sampling of the gut lumen contents. Other mechanisms of antigen transport include specialized populations of macrophages, which you can see in the bottom left panel, and these will squeeze between enterocytes to capture soluble antigens from the intestinal lumen, then internalize these antigens and transfer them to DCs that are in the lamina propria. In the middle panel, you can see goblet cells. This is a cell type whose primary function is to produce mucus, but they can also transfer antigen using transcytosis to the lamina propria. Lastly, on the bottom right panel, some dendritic cells can migrate into the epithelium, where they are referred to as intraepithelial DCs. These DCs will face the intestinal lumen and capture antigens before migrating back into the lamina propria. The dendritic cells of the intestinal mucosa can acquire antigen by any of these methods, and then migrate to the GALT to prime T cell responses. Soluble antigens can also be transcytosed to the lamina propria, where they are then drained to cells in various GALT structures. All of these mechanisms function similarly in that they allow the mucosal immune system to sample antigens from gut lumen. But again, keep in mind that enterocyte apoptosis and cross-presentation of antigens from a dying cell to dendritic cells is kind of the only one that's associated with an active infection. Another unique feature of mucosal surfaces is their ability to secrete IgA isotype antibodies into the mucus layer as well as the organ lumen. If you remember back to our week 5 lectures on antibody functions, IgA is the predominant isotype that is associated with protective functions at all mucosal surfaces. In the gut, these antibodies are produced by plasma cells in the lamina propria, and they also form IgA dimers. However, once they're produced, this IgA dimer then needs to get from the lamina propria into the intestinal lumen. This requires the activity of something called the polyimmunoglobulin receptor, or PIGR, shown in this diagram as the orange rectangle. This molecule is constitutively expressed on the basolateral surface or the basolateral side of enterocytes. It binds to the center of the IgA dimer, internalizing the molecule and then moving it to the luminal side of the epithelial cell through this it's also transcytosis, but it's kind of moving in the opposite direction, as we just learned about with antigen transfer. Once the antibody dimer is moved to the lumen, it is released from the epithelial cell through proteolytic cleavage of the PIGR. This proteolysis event leaves a small portion of cleaved PIGR in complex with the IgA dimer, and this is called the secretory component, and it's shown here in the right panel in orange. This fragment actually protects the IgA dimer from further proteolysis, and it also binds to carbohydrate moieties in the mucus, and this helps retain it in the mucus layer on top of the epithelial cells. Once this soluble complex is released from the cells into the mucus layer, it is referred to as secretory IgA. As a side note, IgG antibodies can also be secreted from mucosal surfaces using this same process with PIGR, but this example focuses on IgA simply because it's the primary antibody isotype that's involved in mucosal immunity. IgA can act in a number of ways to protect the host from invading pathogens by acting both as a secreted form in the mucus, as well as during its transport from the lamina propria into the gut lumen. We've already heard about some of these functions during our Week 5 lectures on antibody functions. In the left panel, we see dimeric IgA binding to pathogens or toxins found in the gut lumen, and in this case, IgA functions by neutralizing harmful molecules in the gut. In the next panel to the right, we have an example where IgA performs similar neutralization activities, but this time it functions intracellularly. So when IgA is internalized by epithelial cells, it can bind to toxins present in epithelial endosomes, and again, the antibody dimer functions here by the process of neutralization. In the third panel, we see luminal IgA binding to antigens on the basolateral side of the epithelial surface. This would be the case if a pathogen or toxin had successfully invaded into the gut and entered the lamina propria. Here, IgA secreted by plasma cells would bind to the pathogens or toxins in the lamina propria and then undergo transcytosis through the epithelial cell to basically spit it out from the mucosal surface into the lumen, where it's exported as an antigen-antibody complex. We can think of this as yet another form of antibody neutralization. Lastly, in the fourth panel, IgA dimers can be internalized by M cells as antibody-antigen complexes, which undergo transcytosis to the lamina propria, where they are taken up by dendritic cells. This process of antigen sampling requires M cell expression of this protein called dectin-1, and again, it's dependent on the binding of IgA to cognate peptide. Note that this is distinct from both M cell transcytosis of whole pathogens, which doesn't require IgA, as well as Fc receptor-mediated transcytosis by epithelial cells. Collectively, all of these mechanisms outline how IgA promotes protective immune responses at the mucosal epithelium. At this point, we've summed up how the mucosal immune system promotes host defense from pathogens. However, I want to end today by talking about a really interesting biological problem, or conundrum, that you find at these mucosal surfaces. And this conundrum is based on the fact that the mucosa are populated by large communities of commensal microbes. This figure from the textbook summarizes the composition of commensal microbiota found at various mucosal surfaces in healthy humans, and this focuses on bacterial phyla, although there are also viruses and fungi that are found at all of these tissues. The commensal microbiota are highly diverse communities that live in symbiosis with the host and carry out several functions that promote host health. They play critical roles in nutrient processing that affects host metabolism, and commensals also take up space and resources that effectively compete with any invading microbes. Intestinal commensals also influence immune development early on in life in a way that affects the immune system systemically beyond just gut-localized responses. And this is through some complex processes that we won't have time to cover in this course. However, I just want us to appreciate the fact that commensal microbes at mucosal surfaces are extremely important in regulating numerous physiological as well as immunological responses in a healthy human. And this is highlighted by many different types of diseases or pathologies which are driven by an imbalance of commensals. And this is sometimes referred to as a state of dysbiosis. It's interesting to consider that most of these beneficial microbes still express things like PAMPs that are shared with pathogenic species of microbes, which should presumably trigger innate immunity through pattern recognition receptor activation. So how does the mucosal immune system differentiate between beneficial commensal species versus pathogenic microbes? One mechanism of tolerance is related to the polarization or activation status of APC subsets found in the mucosa. Unlike other macrophages, lamina propria macrophages primarily function as scavengers that phagocytose debris but don't really process it for antigen presentation, they don't produce inflammatory signaling molecules, and they don't produce high amounts of reactive oxygen or nitrogen species. Instead, these macrophages constitutively produce large amounts of the immunosuppressive cytokine IL-10, which inhibits inflammatory activation of other immune cells, including T cells. These macrophages are therefore said to have a more tolerogenic phenotype, which promotes peripheral immune tolerance against phagocytosed debris. Lamina propria DCs are also interesting in that they can still take up antigen and migrate to GALT to prime T cell responses. but they only do this in response to antigens that are sampled in a way associated with invasive microbial infection. So if they acquire antigen through some of the passive mechanisms that we just learned about, then these DCs tend to not undergo full inflammatory activation. Therefore, lamina propria DCs can exhibit classic inflammatory activation phenotypes when these are needed to respond to pathogens, but at baseline, they tend to stimulate more tolerogenic responses. This panel on the left shows an example of transcytosis of commensal microbes from the gut to the gut lumen to lamina propria DCs. If you remember back to a few slides ago, these M cells transport soluble antigens or whole microbes to DCs, and this occurs even without active microbial invasion of the M cell. There are several other methods for sampling the microbes in the gut lumen like this where they haven't actually infected epithelial cells, and this has important implications for downstream immune responses. Because there is no active infection in this setting, antigens made available to DCs by M cells are phagocytosed under non-inflammatory conditions. So the DCs do not undergo the traditional inflammatory activation that we've learned about so far in this class. These DCs instead trigger a more specialized immune response that primarily consists of low-level IgA secretion from local plasma cells, which is then transported into the gut lumen. This IgA then coats the gut commensals, which limits their ability to adhere to the gut epithelium. Instead, the commensal microbes are retained in the mucus layer, where they can carry out beneficial functions without risk of adhering to and potentially invading the gut epithelium. And this is really a sort of homeostatic state of symbiosis that's found in the healthy gut. A pathogen that actively is trying to infect the host must be capable of accessing the epithelium despite the barriers of mucus and IgA binding. And once cells are intracellularly infected, they can trigger appropriate inflammatory cues to either activate DCs towards a full inflammatory phenotype or trigger the activation of intraepithelial leukocytes for targeted killing. This slide is a little tricky because up until this point in this course, we've mostly associated APC activation with inflammation and initiation of inflammatory adaptive immune responses. However, mucosal immune responses are special in that there are some alternative forms of activation for APCs, and that these suppress inflammation in order to avoid detrimental or negative immunopathologies that you might observe at these epithelial surfaces. This concept of tolerogenic immune responses is important, so I want to end today's lecture by looking at this table from the textbook that summarizes some of the key differences between protective inflammatory responses that take place at mucosal surfaces versus tolerogenic responses. So, protective immunity must be employed against invasive microbes that are capable of overriding the barriers of mucus secretion, antimicrobial peptides, and IgA binding, and this needs to be done in order to successfully infect mucosal epithelium and underlying tissues. In this situation, the body needs to mount the traditional types of inflammatory immune responses that we've learned about in this course. Today, we learned that this type of immune response can be stimulated when antigen is sampled in a way that indicates infection, and that this leads to inflammatory APC activation and stimulation of adaptive responses in the GALT. We also learned how IELs can exert cytotoxic effects immediately within the mucosal epithelium. These promote, again, immediate defense. Lastly, we didn't cover this today, but mucosal surfaces are also enriched with tissue-resident memory T cells that can become reactivated quickly in response to pathogen re-exposure, and this provides a sort of enhanced memory response. Overall, these functions of the immune system are very similar to what we've already learned about in this class, in that it generates a robust inflammatory state that's needed to eliminate infectious pathogens. On the other hand, we've now introduced this concept of mucosal tolerance, which is very important given the sheer amount of commensal microbes and food antigens that are encountered at mucosal surfaces such as the gut. We talked today about how APCs in the lamina propria tend to be more skewed towards a more tolerogenic or immunosuppressive phenotype in response to antigens from non-invasive microbes, and how this polarization is important for preventing adaptive immune stimulation against harmless environmental antigens. Breakdown of mucosal tolerance in response to either commensals or food antigens can lead to numerous diseases and pathologies. So, improper activation of inflammatory mucosal immune responses against commensal microbes is something that's implicated in Crohn's disease. Meanwhile, a breakdown in the peripheral tolerance established in the gut against food antigens obviously leads to food allergies, including celiac disease, which is specifically caused by improper inflammatory responses that are mounted against the wheat protein gluten, as we learned about in our Week 9 lecture on allergy. Lastly, I just want to highlight at the bottom of this figure how the mucosal immune system must find a way to balance tolerogenic responses to innocuous substances while still maintaining the ability to rapidly respond to infection by deploying inflammatory immune responses. This balance has been a recurring theme in the second half of this course, as we've learned that many immunological diseases, or in many different immunological diseases, this balance is skewed towards either inflammation or tolerance. To summarize this lecture, we've learned about some specialized properties of the mucosal immune system that enable rapid responses to infection while maintaining mucosal tolerance against harmless environmental antigens. This is important since mucosal epithelium lines all body cavities, which are exposed to the external environment and are therefore most likely to be the site where pathogens are first encountered. These specialized properties include anatomical features of the immune system, unique effector mechanisms of those cells, and specialized regulatory mechanisms that establish peripheral tolerance in the mucosal epithelium. This last point is very important for preventing the development of inflammatory responses against either harmless antigens associated with food or commensal microbes. That's all of the material that we will cover on the mucosal immune system for this introductory course. Please remember that you have 2 remaining lectures for week 10, in which we will switch gears to discuss an introduction to cancer biology and mechanisms of immune evasion, as well as a new class of immunotherapy drugs used to treat cancer.